Zimbabwe, Lesotho, and also Swaziland. Now, the crop is utilized for its medicinal benefits and also as tea infusion. Now, figure 1 shows Jatropha zeeheri plant. Figure 2, it shows the plant at flowering stage. And figure 3, it shows the fruit of Jatropha zeeheri. Now, the roots of this crop are utilized to treat sexually transmitted infections, urinary tract infection, and also menstrual pains, and also to ensure strong fetus development during pregnancy. The leaves of the crop are utilized as tea beverage making in rural communities, and that is where the study is based. Now, following the commercialization of indigenous herbal teas such as rooibos, such as honeybush tea, and also bush tea, they have gained popularity in recent years. And Jatropha zeeheri is amongst those indigenous tea which are utilized traditionally with multiple uses. However, successful domestication of this crop will require improved soil nutrient status through application of NPK fertilizer. NPK fertilizer in tea production, it improves the physical properties and also it enhances the quality of the crop. However, if NPK fertilizer is not applied appropriately, it can be detrimental to the environment and also to the test crop itself. Therefore, it is necessary to optimize NPK fertilizer application rate that will maximize the productivity while improving the tea quality in terms of physical properties. Therefore, determining optimum NPK fertilizer application rate has the potential for successful production of this crop under greenhouse conditions. Now, the aim of the study was to establish optimum fertilizer application rate that will promote physical properties associated with Jatropha zeeheri indigenous tea's high-quality production under greenhouse. The objective of the study was to assess whether different fertilizer application rates will optimize yield parameters of Jatropha zeeheri tea under the said conditions. Now, in achieving the objective, the study was conducted at Aquaculture Research Unit at the University of Limpopo. The spacing between the plants was 0.3 meters by 0.3 meters for both inter and intra-row spacing. Now, the study comprised of 6 treatments, namely 0, 2, 4, 8, 16, and also 32 grams of NPK fertilizer mixture, which was arranged in a randomized complete block design with 5 replications. Now, the study took 130 days to flower. These are the parameters which were collected during the study: the vine length, stem diameter, chlorophyll content, number of leaves, the leaf width, the leaf length, dry shoot mass, and also the dry root mass. The treatment was applied 7 days after transplanting the seedlings. The seeds were collected at Kureng village. Now, after compiling the data was subjected to analysis of variance procedures using statistics 10.0 software, and the treatment means were separated using Fisher's least significant difference test at the probability level of 5%. And then the significant treatment means were subjected to the lines of best fit, only for those parameters which were significant. Table 1 shows the partitioning mean sum of squares for the mentioned plant parameters. And if you can take a look at the treatment, the one that are highlighted in blue, the chlorophyll content, leaf width, and also the dry root mass, they contributed 70, 78, and 62% in total treatment variation, and they were highly significant. And the plant parameters in black, they were significant. And vine length, stem diameter, and leaf length, they contributed 47, 60, and 49% in total treatment variation. And the one highlighted in red, they were not significantly affected by the NPK fertilizer. Now, the two graphs shows the response of the said plant parameters to increasing rates of NPK fertilizer under greenhouse conditions. If you can take a look at the two graphs, it shows that the plant parameters or biological indices, they were responding to increasing concentration of NPK fertilizer rates. As shown in low concentration, there was a bit of stimulation, and at high concentrations, there was the treatment was phytotoxic to the plant variables. This is shown by the density-dependent growth patterns, which are observed in the two graphs. Similarly, in figure 7, the response of chlorophyll content and also the leaf width explains the same. And also the leaf length and dry root mass, they've shown that the response to increasing concentrations of NPK fertilizer rates, and also the more you increase the dose, the more they are phytotoxic to the test crop or plant variables. Now, looking at table 2, it shows the quadratic relationship, the coefficient of determination, and also the computed optimum of NPK fertilizer rate. If you take a look at the coefficient of determination, which is shown by the R squared, it shows the strong relationship between the plant variables and also the NPK fertilizer rates. And then the X there, it shows the optimized application rate, fertilizer application rate for the mentioned plant variables. And then the optimum NPK fertilizer application rate was found to be 3.34 grams of NPK fertilizer. Now, in conclusion, Jatropha zeeheri requires low quantities of NPK fertilizer for increased plant growth, and also it can be supported by the fact that the crop is indigenous to South Africa. And also, the density-dependent growth patterns were observed in the current study, whereby stimulation was observed at low rates of NPK fertilizer. In contrary, the NPK fertilizer at high concentrations or application rates were phytotoxic to the test crop. NPK fertilizer for Jatropha zeeheri was optimized at 3.34 grams per plant NPK mixture, which can easily be translated to 33 kilograms NPK mixture per hectare. It can be concluded that Jatropha zeeheri appears to qualify as a low input tea crop. I would like to acknowledge the University of Limpopo for allowing me to conduct the study, NRF and TIA for funding. Thank you.
Thank you very much, Happy Bango, for the presentation. Shall we have the questions and comment for Happy?
Thank you. The first thing, this is a comment. You did mention that NPK determines the physical properties of the soil. Is that correct?
Pardon?
Did you say NPK determines the physical properties of the soil?
No, it improves.
It improves?
Yes.
Okay. The other thing is, you indicated that your experimental design was RCBD, right?
Yes.
But when you performed your statistical analysis, I didn't see any block there.
It is there on table.
I looked at I saw the replication, I saw the treatment. I didn't see the block. Maybe I missed it.
The block is the replication.
No, it's not. It can't be. If you look at the degrees of freedom, it tells you it can't be. So, but anyway, if you look at it, it can't be the same thing. Your replication and your block are two different things. I want you to look at that analysis again.
All right, I'll
and
look at it.
make sure you incorporate both. Actually, it's for the other guys who have got a similar situation. I mentioned it before. If when you are doing this thing, it's best to write your linear model to show what factors are on the right-hand side. That way you know exactly what you have done,
Yeah.
whether it tallies with the experimental design which you gave.
All right.
Or maybe you can answer in this way. What was your blocking factor? What were you blocking in this study?
I was blocking against the unequal distribution of sunlight, which is caused by the tree that is grown next to the greenhouse.
And I want to see the blocks in that model, because if there is differential performance due to the aspect of the sun,
Yes.
you have to take that variation out. Otherwise, if you don't, it's going to go into the error model or error part of the model.
Agreed.
So you have to have the block there so that the variation due to the distribution of the sun is not thrown into the error term of your analysis.
Agreed.
It will actually help you out, because once you remove that, you might pick up significant differences where you could not, because your error term is inflated by leaving the block out.
All right, thank you.
Okay.
Thanks very much, Mr. Bango. I have 4, but I'll try to reduce them. 1, on your treatments, you have 0. I understand you want to have a negative control, but don't you think you are starving the crop if you are not putting any fertilizer on it, and you're comparing it with where you put, for example, 4? That's the first one. And the second one, I want to know, because I saw that you did analysis of variance, how did you determine that your data were normally distributed? And also, which model did you use to optimize the concentration?
Okay. All right. The first question. The reason why I used 0 as control is that the 0 is for, I was applying water only. There was no treatment at all. So that I can compare how the plants have performed when they are exposed to increasing NPK fertilizer rates. So it was meant for comparison, so that we can see what effect does the NPK fertilizer has on the crop. And then the second one, I used the quadratic relation model, which shows the plant variables and also their sensitivity to the NPK fertilizer rate. Thank you.
Just the first one. To come to what he asked, I wonder, in terms of ethical understanding of the study, you cannot put 0 there. I mean, the results would be obvious. Secondly, you have mentioned that when you increase the rates, it tends to increase the yield, and then it goes down. What reason would you give for it going down? The reason I'm asking this question is there are a number of elements in NPK, and you can't attribute any toxic effect on any particular one. So how Could you explain your toxicity, as you said?
Okay, thank you. Thank you, Prof. It has been shown with the results that, as you were saying, that as I was increasing the dose, the plant variables were phytotoxic. I mean, the NPK fertilizer rate was phytotoxic to the crop. If you take a look at the functions of NPK, the nitrogen, I mean, your phosphorus and also potassium, you will realize that if you put more of potassium, I mean phosphorus, you will result in your crop being burnt out by the increased dosage of the NPK, I mean the N fertilizer. However, the growth will be stimulated. The plant leaves will be stimulated, but the roots will be affected. So many farmers abuse fertilizers. So that is why we must find an optimum fertilizer rate that will promote the crop itself while not affecting the environment where it is grown. Thank you.
Thank you very much, Happy, for the presentation and the contribution. Can we give Happy a round of applause as we call upon Isaac Litswalo? Isaac, are you in? Isaac is also a staff member who's going to present on the effect of mulching materials on growth and yield of tomato, a perspective of climate-smart agriculture. Over to you, Isaac.
Okay, ladies and gentlemen, good afternoon. I believe it's, good afternoon. Please allow me to present to you some research that was consolidated, entitled the Effect of Mulching Materials on Growth and Yield of Tomato, which was a perspective of climate-smart agriculture. So this work was under the concerted efforts of myself, the underlined name there, and Dr. Sateche. Just to introduce and also to bring to your attention what was happening. So obviously I'll be talking about the tomato, which is a consumed vegetable crop worldwide because of its nutritional properties. So this, just to elucidate on these nutritional properties, this is because it has some good properties like phosphorus, vitamin A, and vitamin C. However, with those important things mentioned, its yield is very much limited, and simply because if you check the province that we live in, it is water scarce, it is a dryland area, so there is a need for us to look at some innovative practices that will cap for that problem. So the last point there, we'll be talking about the mulching, of which it is the driving factor in this work. So normally this mulch, it enhances yield and also growth of tomato. In recent literature that was discussed, hence there are other different types of mulches. You look at the likes of biodegradable mulches, degradable, organic, and inorganic mulches. So now, beside that, looking at the real problem, why did we have to come up with this information? So we looked at it this way. Ever since we have a low rainfall in Limpopo province, we found it very much wanting to say, let's look at what we can do in order to conserve moisture and also to control the weeds, of which weeds infestation are also a problem. Then, looking at drought stress in tomato, it is not, let's not underestimate the fact that tomato is drought susceptible. So, which means mulching materials, that of which I'm going to show you, are very much important when we want to look at moisture conservation. So this study aimed at investigating the effect of those mulching materials on growth and yield of that tomato. Then the rationale behind this is that tomatoes, they do play a crucial role in the health, human health in South Africa, nutritionally and also economically. So when we look at the nutritional part of it, I also mentioned in the introduction, like it has vitamin A and C and also phosphorus. And then their production is also threatened by climate change. So this was also addressed in the previous problem statement that I was discussing with you. Then the third point, mulches of various types, they also play a role in mitigating against the negative effects of extreme weather. So, as I have mentioned that we are living in a dryland area, that is a problem, it's a negative setback that we really wanted to look at. However, there is limited information on the use of organic mulches. Okay, so the tools and also the procedures that helped us to come up with some sort of information from this. I will start by the site location, which was GBRCE, this at University of Limpopo. Then the procedure there, the soil was pasteurized for an hour at around 360 degrees. And then we also fumigated the plot area to remove any other factors that will tamper with the treatment, our treatments. And then we transplanted the seedlings containing hydromix and also sandy loam soil with a 3:1 ratio. Then crop management and the spacing. So this crop management, I want to bring to your attention about the irrigation. So we were applying water 250 mils each and every second day. And in relation to pests, the pests that were identified was the leaf miners and whiteflies, of which we used cypermethrin to control those insects. And also maybe nutrient supply. We did apply multi-feed as well, considering the fact that we have also pasteurized the soil, which means we have also tampered with the nutrient available in that particular soil. The treatments, we had different types of mulches. Okay, I jumped this one. The fifth point, we had no mulch, pine bark, sawdust, and black plastic. Those were the mulching treatments that we had. And we used a randomized complete block design with 4 replications. The data were collected at pre-harvest and at maturity of the crop. So these are the particular variables or other parameters that we have collected: chlorophyll content, plant height, number of primary branches, days to first flowering, and at harvest, number of fruits and fresh biomass. Then, the data, after collecting some of this data, we subjected it to analysis of variance using GenStat 20 version, and we used least significant difference just to pinpoint the treatment effect or the treatment difference that arises from that at 5% probability. So these are the results that we got. And the first variable, chlorophyll content, we did not find any significant differences, which means whether you apply mulch or not, you will still have the same values, or the chlorophyll content will not change. And then as for the plant height, we had some significant differences. So if I look at, I'll just pinpoint the one which produced the highest, because in real life, when you want nice things, you'll just go for them. You don't hesitate. So I'll just go straight to pine bark, which produced the tallest plant height.
2 minutes left.
Thank you. Compared to the plastic mulch. Okay. Then, number of primary branches, there was also significant differences, as evidenced by the small letters on top of each and every bar, knowing that the different letters means significant differences. Days of flowering, there were no significant differences. Okay, and then number of fruits, pine bark also produced the highest number of fruits compared to the other mulches, and there were significant differences. Then, fresh biomass there was also found to be significant. Then, the concluding remarks, take-home assessment or activity. Then, we found that various mulching materials could be used as climate-smart practice in tomato production, because we had some information that was new and that could help us. Pine bark improved growth and yield of tomato. Then, these materials could be used to tomato farmers in general, because you don't buy them, you can just go and then you just load and then you apply. Thank you.
Thank you very much, Isaac Lizolo, for the presentation. Shall we take the questions and comments for Isaac?
Thanks very much, Mr. Lozolo. I must indicate that I'm also part of the work, but for education purposes, I want you really to somehow educate us. Based on your results, I saw that sawdust had sort of a negative effect compared to the pine bark. Do you have any reason why you think sawdust performed bad?
Okay, thank you for that. That is a very good question. If I may just compare, pine bark, it has some polyphenols and phenolic compounds that are evidenced to increase root growth. Then on the contrary, looking at sawdust, I don't want to say something that will impose another factor inside, but I'll just try to, Sawdust is very light, and it doesn't have more weight. And just to add on that, it has more tannins, and it also has crude fiber inside. So that could lead to those negative side effects. Thank you.
Okay, it looks like everything is clear, or either the energy levels are too low. But can we give Isaac Lizolo a round of applause? Okay, thank you very much for being the lovely audience for the first afternoon. I must apologize to Kulufelo Lezong. We were supposed to have your presentation now, but as you can see, the ripple effect of the technical glitches that we experienced, but also the slower start in the beginning. We are going to start with you after lunch. I hope, Kulufelo, you understand that. We'll start with you in the afternoon session. We are going to now take a break, go and have lunch. I'm told is at Limpopo Hall. Also to try and circumvent the challenges of time and also the load shedding challenges that we're experiencing, can we just agree to take our lunch for 45 minutes? May I also ask the presenters in the afternoon session to come and load the presentation so that we don't take a lot of time trying to work on the presentation. As the rest of the audiences are going for lunch, can we have those that are presenting in the afternoon to come and upload the presentation? Thank you very much for all the discussion, and we are looking forward to see you in the next round of the session in the afternoon. 45 minutes strictly. We will come back and we will start. Our adjudicators, thank you so much. Please go and have lunch. Strictly after 45 minutes, let us be back. Once we have the presenters and the adjudicators, we are going to continue with the session. Thank you. They didn't say about it. I don't know who to ask now. Because there's